Dear class 10, we will continue chapter number 8, that is the circulatory system. In the last session, we discussed about RBC or erythrocytes. Then in the last virtual class, and then in the last live session, we discussed about second type of blood cell, that is WBC, which are also known as leukocytes. And today in this virtual class, we'll talk about the third type of blood cell called blood platelets. Okay, blood platelets, they are also known as the initiator of clotting of blood. Okay, that means they are the ones which initiate the clotting of blood, because clotting of blood is very important when we are injured. Okay, when some parts are accidentally cut, you know, we know that the blood starts flowing, and after some time it has to be stopped. Okay, so how it stops? Because at that point where it is caught, some kind of clot is formed which blocks the blood from flowing. Okay, so that is all done by the blood platelets, and that particular phenomenon is called clotting of blood. All right, and you see, these blood plates are minute oval or round shaped non-nucleated cells. Okay, non-nucleated cells means cells having no nucleus. Okay, there in the last session itself we came to know that even RBC are non-nucleated, especially during its maturity. Okay, but WBC has nucleus, so they are nucleated. But these blood platelets are also non-nucleated because they do not have nucleus. Okay, and their number, number ranges from 2 lakhs to 4 lakhs. That means, see, the number of blood platelets is found to be ranging from 2 lakhs to 2 lakhs to 4 lakhs per cubic millimeter of blood. Okay, so in 1 cubic millimeter of blood, okay, in this volume of blood, okay, or in 1 cubic millimeter of blood, the blood platelets count is this 2 lakhs to 4 lakhs. Okay, whereas RBC we discussed last time that it is 5 millions per cubic millimeter blood in an adult male, whereas in an adult female it is found to be 4.5 million. Similarly, WBC 4,000 to 8,000 per cubic millimeter blood, whereas blood platelets are found to be 2 lakhs to 4 lakhs per cubic millimeter of blood. Okay, that is the count of blood platelets. Then place of formation, where are they formed? So they are formed there in red bone marrow itself. Okay, they are formed there in red bone marrow itself, and they are derived from the giant cells of bone marrow, red bone marrow. Okay, and which are known as megakaryocytes. Okay, megakaryocytes are the giant cells of red bone marrow, where from which your blood platelets are derived. Okay, lifespan. So how long do they survive? The lifespan is 3 to 5 days only. Okay, only 3 to 5 days. Okay, RBC survives for 120 days. Your WBC 2 weeks, whereas this your blood platelets survive for 3 to 5 days, and after that they are destroyed there in spleen. Okay, spleen, which is an organ of lymphatic system. Okay, now we'll talk about the function of these blood platelets. We have already come to know that it is the initiator of clotting of blood, which means it initiates the coagulation of the blood. It has to be clotted when the injury take place. Okay, after the injury, the blood has to be clotted, then only it can be stopped flowing. Otherwise, what happens is that the blood keeps flowing. That means it bleeds to death. Person will die because of the loss of the blood, no matter how small cut it is, how small injury it is, if it is not clotting. All right, so that clotting of the blood is very important, which is mainly because of the role being played by blood platelets. Okay, so how does this clotting take place? Let's see here. So it takes place by a number of chemical reactions there. So the chemical reactions have been shown out here with this schematic diagram, in fact, with this flow chart, which is also given there in your book. Okay, so main reactions in blood clotting. So first, what happens? Injured tissue cells and platelets. That means your blood flows from where and when? When there is an injury, when some parts are caught. Okay, so that time the blood flows from that region where the injury has taken place. Okay, so when it is injured, that time what happens? The tissue cells or the platelets get damaged. Okay, they get what? Damaged. They are injured. So when they are injured, that time the platelets, okay, these platelets release a kind of enzyme called thrombokinase. Please see the spelling, thrombokinase, which is also known as thromboplastin or factor X, or it is even called Stewart factor. Okay, it's a kind of enzyme. All right. Now this thrombokinase, which is an enzyme, okay, with the help of calcium ions, okay, with the help of calcium ions, convert this inactive prothrombin, a kind of substance is there which is called prothrombin. Okay, at this form it is inactive. Okay, so thrombokinase with the help of calcium ions converts this inactive prothrombin into active thrombin. Okay, prothrombin itself will be converted into thrombin. The difference is that it is inactive, now it is active. Okay, remember prothrombin, okay, requires a kind of a vitamin, that is vitamin K. Okay, fat soluble vitamin. So that vitamin K is required for its formation. That means vitamin K is important for the formation of this prothrombin. Okay, so this inactive prothrombin, which is present there in blood plasma, will be converted into its active form called thrombin. Okay, then what happens? This now active thrombin, again this active thrombin with the help of calcium ions convert a kind of protein, okay, a kind of protein present there in blood called fibrinogen, which is present there in blood plasma itself. So fibrinogen, which is soluble, okay, into the insoluble form called fibrin. Okay, similarly here you see thrombin with the help of calcium ions converts soluble fibrinogen into insoluble fibrin. Okay, last time we were talking about the this blood in its introductory class, in its introductory session. We have discussed about this fibrinogen and all. So here you see this thrombin with the help of calcium ions converts this soluble fibrinogen into fibrin, which is insoluble. Okay, then what happens? This fibrin now forms a network of sticky threads. Okay, being exposed to the air. What happens? That your fibrin now forms a network of thread, forms a mesh of thread. Okay, and which catches RBCs also now, which catches that erythrocytes there, then gets slowly solidified, which is called clot, and it is also called thrombus. So this is how the clotting of the blood takes place. Okay, so now when the clot is formed there, at that point where it is caught, will block the flow of blood. Okay, then beside that clot there is a pale, fluid substance is seen. Okay, pale fluid substance is seen, which is called serum. Okay, it's called what? Serum. So that means last time we discussed plasma minus fibrinogen is called serum. Plasma, okay, minus fibrinogen equals serum. Okay, it was discussed last time also. Okay, this fibrinogen. So that's a protein. When it's removed from plasma, it becomes serum. Same thing is happening here also. So clot, beside the clot there you see a pale yellow colored fluid substance is seen, which is called serum because there is no fibrinogen. Okay, fibrinogen is not there, which is already trapped there in the thread. So this is how the clotting of the blood take place, which is all because of this particular blood cell called blood platelets. So that they are also called the initiators of clotting of blood. Okay, now we'll talk about the disease related to blood platelets. So first comes here hemophilia. So hemophilia is a genetic disorder. Okay, genetic disorder. Genetic disorder means if the parents have this problem, the children also are likely to have this problem. Okay, so this is called genetic disorder. So hemophilia is also genetic disorder. During which what happens is that the blood does not clot properly due to the lack of blood clotting protein. Okay, blood clotting protein is your fibrinogen. Fibrinogen. Okay, if there is no fibrinogen in the blood, then that problem will be there. Okay, and similarly, when the number of blood platelets also falls abnormally to very low count at the time also, the blood does not clot properly, okay, leading to the hemorrhage. All right, and even in some problems like dengue viral fever. Okay, dengue viral fever. Okay, even in dengue viral fever, the problem will be the same. Okay, where the this blood platelets count will be very low. Consequently, the person will have that problem, which means clotting of blood problem will be there, and consequently the people might die. Okay, so here in India also, especially in Delhi and other states of India, so many people have been killed. Okay, so it has taken the hundreds of life since 1996 till date. Okay, so because of this problem only. All right, so today we'll do only this much, and we'll continue the chapter in the other session as well. Okay, thank you very much.